Charles, thanks for sitting down with me today. I appreciate it.
No, my pleasure.
Charles, DNA Landmarks was founded in a partnership or joint venture with SWS and A Canada. Mm-hmm. Can you tell us a little bit about how that started, how it transitioned into being owned by BASF, and what advantages come with that transition?
Sure. Well, actually, the company, before it started, there was a group of researchers at Agriculture Canada, and they were approached by Svalof Weibull and said, hey, we're doing a lot of work in Canada in canola at the time, and we want to do more work in markers. Why don't you leave Agriculture Canada and start a company, and we'll help you set it up, which is what happened. And actually, it was funny because they resigned from Agriculture Canada and then came back the next day and asked if they could rent the lab space back from them. So
Perfect.
That's where they started. But it was only a couple of years after that BASF wanted to get into the plant science business, and they put a joint venture together with Svalof Weibull because Svalof had seed experience. And DNA Landmarks became part of BASF through that joint venture. Since that time, BASF bought out Svalof's share, so it's now 100% owned. We're 100% owned by BASF. So it was a change. It was a dramatic change from 5 people up to 12 or 15, and eventually now where we're at close to 50 over a relatively short period of time. But being part of BASF, there's a tremendous number of advantages. We do work all over the world, and wherever we go, BASF has infrastructure that we can depend on, contacts, networks, and operationally, a company like ours has had to be very, very focused on operations because people come to us and ask us to basically give them do part of the research for them. Right. They provide us with materials that are part of their breeding program, and we give them results that they make decisions on. And so they need to be able to trust us that we're doing everything we possibly can to eliminate error from every process that we do. And BASF has been very helpful with that. They're a very process-oriented company, like most ag chemical companies would be, and that's really helped us quite a bit. We have a lot of very, very high stringency in our processes to get that done correctly.
Speaking of processes, last fall you just announced that you've discovered some new markers for disease resistance in tomatoes. Mm-hmm. Can you tell us a little bit about what's involved in that and what that actually means to the industry?
Sure. To be fair, these markers were around before, and they're fairly basic diseases in tomato, fairly common diseases in tomato like Phytophthora and Verticillium and things like that. What we did though is we converted them from an older technology or an older type of marker, which was, in some cases they were SSR markers, which are simple sequence repeats. In some cases they were CAPS markers. These are markers that are much less efficient to run, much more costly to run, and so it was straining a lot of breeders' budgets, especially some of the medium-sized breeders, to screen on a large scale for these. And we converted them to single nucleotide polymorphism markers, or SNP markers, which is sort of what is the gold standard now in genomic technology. And this drops the cost dramatically from $1 or $2 a marker, we're down to 20 cents a marker or less. So we can run them as a whole suite altogether in a multiplex of 8 or 10 markers, which is really quite cost efficient. Or we can run them individually, but it's still, either way that it happens, it's still a lot less than it was before, and so it's really kind of opened things up.
Speaking of genomics, when we speak as genomics specifically, is there anything that you can point to or give us an example of over the last 12 months that has really been a bit of a game changer as far as genomics in the seed industry are concerned?
I think still right now the biggest thing in genomics from the standpoint of technology is the impact of sequencing technology and how it's evolving. That process, that technology started to change about 5 years ago. And it's one of these things now. It's going faster in some cases than you would expect. For instance, with Moore's law, where we're seeing the amount of output dramatically increasing. This year, one of the companies that we deal with in sequencing is announcing that they're successfully, from a single run out of their machine, you can produce a terabase of data. That's the equivalent of over 300 human genomes in one run. So now, to be fair, when we sequence, we typically do high redundancy, 20, 30 times redundancy, so that we have a high level of accuracy, but it's still a tremendous amount of data. And combine that with sort of being able to target specifically areas that we'd like to sequence, it's really changing things quite dramatically.
12 months seems like an eternity in your business. When you look forward into 2014, what do you see that's got you excited for DNA Landmarks specifically, and then more in general, in genomics in general?
I think that we're still there's still a lot further to go with the sequencing technology. I see like there's a couple of other companies that are bringing new technologies onto the market, which will change things quite dramatically. One of the things that we look at in sequencing, and I don't want to get too technological here, but it's one thing to produce a lot of sequence data. But the question is how long are the reads of those data? So if it's only 200 or 300 base pairs long, it's like putting a puzzle together, and the more pieces you have, the more complex it is. There are some technologies coming out that are going to allow us to do much longer reads, make it much easier to reassemble the genome after we do it. So that's quite exciting. And overall, the cost of the technology is allowing us to open up so we can move into smaller species in some of the vegetable crops, for instance, and stuff like that, which really hasn't been able to be cracked yet, or in some cases tackle much more complex genomes. I mean, wheat is one of the most complex genomes. It's a hexaploid. It's a very large genome, and so there's a lot of sequencing to be done there, and a lot of stuff to figure out. And the more efficient this technology gets, is what allows us to tackle these really important species that have been much tougher to crack than, say, corn and soybean, which I've had a lot of focus on them and are much better studied genomes.
So when we talk about wheat, any vision from you on when we can see that being moved to the next level?
Well, I mean, it's kind of ongoing. There are several groups that are working on wheat, and some are in the public domain and some are in the private. It's hard for me to comment on the private stuff because if we're doing it, we're not allowed to talk about it, and if someone else is doing it, they're not telling us. So, but it is moving forward. And of course, there's been a lot of talk about hybrid wheat, and that's not really my specialty, but I think it's also showing, I think, that the industry is extremely focused on it because, if they can make a hybrid out of it, it improves yield, but it also allows them to capture a lot more of the value, and that then allows them to do the kind of R&D that they're doing on corn and soybean, the large players. And I think that's why you haven't seen wheat. It's such an important crop agriculturally, and of course in Canada, for us, it's very important, but all over the world. But a lot of the big ag biotech companies have not invested into it as heavily as they have in corn and soybean, and I think it's a matter of value capture. So I think that's coming as well.
Again, speaking of value capture, Minister Ritz just made an announcement about UPOV 91. Finally, any thoughts or comments on what that means for, maybe even specifically for your company, and then also for the industry again?
From a standpoint of variety protection and stuff like that?
Canada adopting UPOV.
Yeah. I think it's really, I think it's excellent. I think that as we go forward and things become more and more, companies invest more and more into the varieties they're developing, protecting them abroad is going to become, much more important. And also having that level of confidence in our own system, so that when people can put varieties out anywhere that they want to and know that they're not going to be taken or used inappropriately or anything like that. From a technological standpoint, of course, variety protection has always been something that we've been involved with because markers are essentially a fingerprint, so companies are always fingerprinting their own varieties, checking to see that people aren't encroaching on their genetic material, and all these types of things. They're big parts of patent filings and various things like that. So definitely that's going to be something that we see going forward.
Great. Charles, thanks again for sitting down with me. I appreciate it.
My pleasure. Thank you.